Hello guys, in this video we are going to see the auditory neural pathway and the physiology of hearing. First, the outer hair cells and inner hair cells, out of which the outer hair cells play a major role in converting the mechanical energy that is produced by the sound wave through the fluid, and it converts this mechanical energy into electrical energy. This is carried by the eighth nerve. You can see from the diagram, eighth nerve, from the cochlea to the eighth nerve, this auditory nerve or eighth nerve. From there it goes to the cochlear nucleus, and then the superior olivary nucleus, then to the lateral lemniscus, then inferior colliculus, then medial geniculate body or medial geniculate nucleus, then into the right auditory cortex. You can remember this with the famous mnemonic, that is E. coli mo. The E is in the ear, COLI is present in the brain, and MA is present in the brain. Okay guys, before above the cortex. uh, okay, then the cochlear nucleus is in the brainstem. Crossover takes place at the level of superior olivary nucleus. Then we can measure this with an audiometry known as brainstem evoked response audiometry, or BERA or ABR. It is a very important audiometry test. It is a study of electric waves produced along the neural pathway of sound. Okay, here also you can use the mnemonic double E. There is a single E, but here it is a double E coli, whereas the E or eighth nerve correspond to the wave 1 and wave 2, whereas the wave 2 is produced by the distal part. The wave 1 is produced by the distal part, wave 2 produced by the proximal part of eighth nerve. Then wave 3 is produced by the cochlear nucleus, wave 4 is produced by the superior olivary nucleus, wave 5 is produced by the lateral lemniscus, wave 6 is produced by the inferior colliculus. See, proximal part of auditory nerve, distal part of auditory nerve, then cochlear nucleus, then superior olivary nucleus, then lateral lemniscus, then inferior colliculus. The 5, 6. Then the wave 5 is the largest or tallest or most several or most significant wave. So you can see is the wave 5. It is a very important wave. It can tell whether there is a problem is due to cochlear or retrocochlear cause. See, then we need to see about the interpretation of this BERA. If there is no wave, or any waves are not present or completely absent, it is known as based as cochlear loss or sensorineural hearing loss. Total sensorineural is lost. If wave 1 is present and wave 5 is absent, that means the cochlea is normal, but there is a problem behind the cochlea or retrocochlear hearing loss. Okay, and even if there is a delayed, normally the distance between the wave 1 and wave 5 is 4.2 seconds. If it is more than 4 seconds, more than or equal to 4.4 seconds, it tells that there may be retrocochlear hearing loss. The first wave is normally headed 1 to 1.5 seconds. That means the cochlea is normal, exactly normal, so we don't need to fear about that. This BERA is a confirmatory test for neonatal deafness. The neonates can't express their deafness. So we need to conduct this BERA and we can tell there is a deafness or not. It's a very specific test. It is also used to detect malingering, whether a person is saying that he is truly a deafness or is malingering. BERA is an objective test. Okay guys, thanks for the video.